Okay, thank you, David. Hello, everyone. I'm Yuqi from Duke Radiology, and I will present a method that offers a rapid and processed way to count cell phenotypes across various brain regions. And let's go ahead. Okay. So many experts at University of Tennessee have worked on an established histology method, which is a well-recognized and a rigorous way to count the cell phenotypes. And for those who are unfamiliar with the terminology, histology is a systematic approach that involves meticulous chemical processing and manual counting under the microscope. And our method is our report here is built upon the histology and allow us to flexibly work on different brain regions rapidly. And the pictures I showed here indicate that the cell phenotypes at different brain regions can have different features, and that requires our method to be able to flexibly fit to different brain regions. And the first step of the method is to correct the morphology of both the brain label atlas and the microscopy images. We acquire the image of the magnetic resonance histology when the brain sample is inside the skull, and we acquire the microscopy images when the brain is taken out of the skull and being chemically processed. So this step is to ensure that our results will not be influenced by the distortion in the brain label atlas and the microscopy images. And then I will briefly introduce the methodology part. It starts from the geometry distortion correction based on the MR histology, as we just introduced just now. And the actual pipeline of the algorithm start from the segmentation of the brain region based on the user-defined label. And then for each brain region, this algorithm will generate a user-defined number of the subvolumes, and the subvolumes generation is to reduce the computational efforts and the memory used to save those files. And then for each subvolume, a light-weighted supervised classification algorithm, which is random forest here, will be applied to the subvolume to generate the blob of the nested neurons from the background. And then these blobs of the neurons will then be sent to the post-processing part of the algorithm, which include the three-dimensional watershed, the volume filter, and the unique blob labeling. And because the sub-volume are user-defined, so our algorithm will actually output the density instead of the counts. And then the densities will be saved into a files. Okay, and then the only manual part of the whole algorithm is the random forest training. And this step will take less than 1 minute. And as shown in this panel, that when there are about 20 identified neurons that will be input to the training, and then the F score of the validation will be around 0.9, which is already quite high. So this step kind of ensure our algorithm to be easily adapt to different regions and translatable across regions and specimen. And here is some quantification about the results. This one shows the comparison between our result and the existing literatures. The circles here indicate the density, the converted density from other literatures, and our results are shown as a bar here. This panel shows that our results are mostly consistent with other peer results. And if you want to know more details, please refer to our previous papers. And thank you.